Okay, we're going to have two fantastic guests. David Cappaert is an entomologist and educator with Quamash Eco Research. He has a research background in biocontrol and pest management. His current passion is bees, and he has a particular fondness for Andrena. And he's talked about Andrena before. You can go watch that YouTube. And Amanda Konishi is an artist and scientific illustrator. Her work focuses on honoring the small environments around us, with an interest in community engagement through the intersection of art and science. And the topic is, oh, it's a long one, bee glossary, an entree to the language of bee taxonomy. 50 years ago, the identification of bees required fluency, still does actually require fluency in morphological terminology. You would need to recognize and evaluate the propodial carina, ochraceous coloration, or rugulose surface texture. In recent years, and with the rise of citizen science, there are better tools written and illustrated with non-specialists in mind. But to navigate most keys still requires bee-specific terminology and a working knowledge of how to apply terms across taxa. So David has developed a glossary of terms that describe bees, a guide for the non-taxonomist, and they will share the story of why they built this and who might benefit and how you can contribute, which I've helped out a little bit. It's been really fun to do. This will be the debut of Amanda's illustrations, which anchor the glossary. So David, are you ready to take over?
I am quite ready. I guess I sent you that really long intro, and now it almost obviates the need for this whole talk, but let's do it anyway. So,
you got one. Thank you.
And if I can get through this like reasonably quickly, I'd be very happy to have questions or ideas from this group. So one of the problems, if you're like me, I'm a retired person, and I like the closest I come to interacting with people is what we're doing right now, but I mostly sit at my desk. And so I don't have any great way to drag people in from the office next door and ask them, like, what do you think about this? So this group is going to do that for me, I hope. So this is about this big epic project. Here's our names here. I guess you've already done that intro. We're going to see these drawings. We'll play with these a little bit later. But one of the beautiful things about these, I don't think there's anywhere else on the internet where this can be found. The little labels for each one of these views of a bee are hyperlinked, so you can just jump to a piece about mesosoma and learn more about that if you want to do it. So that's the general structure of it. And so what I want to just walk through is why I did this, who do I think will benefit, how did I put it together, and Amanda has her piece. It's not straightforward to make an accurate and beautiful and morphologically precise image. She'll tell you how that all happened. And then, maybe some ideas about what we have now and what else should I do with this thing. And I asterisk here that I use I because I am sole author, and not because that's how I wanted to do it necessarily. I just didn't get anybody else who jumped in. People said it's great, but yeah, I did all the grunt work. So I own all of the mistakes. So why for me, it's pretty easy. Like, bees are really popular now. And that's your group, 10 years ago, there wasn't a group like this in Washington. And now there's got a huge membership. And it's true for, all across the country. And a good example would be all the different sort of bumblebee citizen science projects. Bumblebees are an interesting special case because you kind of don't need a microscope to figure them out. And so a lot of the stuff that I'll talk about in the glossary is, you don't need it for bumblebees so much. It's about colors and, a few things that you might measure. So anyway, so we have bumblebees. One, really good and quantitative way to look at citizen science as respects bees is in iNaturalist. And I've seen a number of the people who are in this group on iNaturalist. It's a really good way to trade things back and forth. And you can easily query the database and determine that there's 70,000 bee observations in the state of Washington. 8,000 people have contributed those, 300 species. So it's a really impressive thing. Although I asterisk in this case that I'm sure anybody who engages with iNaturalist is aware of this limitation. It's hard to be rock solid on an ID from a fuzzy picture in this app. So lots of those 70,000 observations, they're great, but they're not research grade. So they haven't been sort of verified by people who really know. And a lot of them are also not to species, because it's not hard to say it's an andrina. It's really hard to say which species that is. And then I just, for fun, I grabbed a couple of the people who I know might be here, who are from Washington. And for me, this is an example of, like, on iNaturalist, this, pardon me if I got this wrong, Carla, but this would be clearly someone who's not just snapshotting whatever landed on the gardenias, but wants to really figure out what the heck this is. And you can tell that by the numbers of photographs that are taken, and then it's a bee on a pin. And so if I could click on these, this isn't live, but you'd be able to see she tried to show any number of views. And it's the fun thing about iNaturalist is that Carla can say what she thinks it is, and then she says, "Please, August Jackson, will you tell me what you think?" And August is like, one of the very best. And so if he says, "Yeah, I think it's this species," that's a gold standard, I think. And then just toss one out here for Lisa too. And it was just interesting because I grabbed this sort of, also because I thought it was a good picture, also because I thought it was nice to show that Lisa knows you want to get 5 views of that one insect if you possibly can, because the features might not be visible in one. And then she gets another opinion from Swiftchick, i.e. Carla, who was in the previous slide. So this all gets, I'm not going to say incestuous, it gets, intimate in terms of who's out there and who's paying attention to what you do. So anyway, I'm really jazzed about iNaturalist, but I'm going to tell you why I'm going beyond that. Bees are hard, right? So iNaturalist is very good for butterflies. You can't get it wrong, and they're big, and you can get what you want. But for a lot of the bees, if you want to know species, you're going to have to look at little bugs on pins and really look at them and think a lot about it, and consult a lot of resources. So, oops. So here is, I took this example, Osmia atrocyanea. It's one of my favorites. And it happens there's only 3 named to species in iNaturalist out of those 80,000 observations. There's just 3 for this. 2 of them are mine. And then they're there because, I mean, you can see they're all on pins. So I knew, and whoever else submitted one here, knew that you really have to try to get some details in there. You have to be able to show the different views and hopefully at pretty high res. But how did I get to atrocyanea in the first place? I did the hard stuff that you have to do if you want to get to species on a lot of these different little bugs. So if you want to key something out in Osmia, you need to look at the punctation, how the pits are distributed and what they're next to, and the size and the shape of the teeth. It's huge for Osmia, and really aggravating because they close their little jaws and you can't see their teeth a lot of times. So it's really tough. But let's say I decided that I needed to figure out this Osmia, and I photographed it, or I just looked at it under my microscope. I'm going to try to find some resource that I can rely on to key it out. And one might be, there are other options, but there's a lot of times when you do need to go to something like 1939 Sandhouse. So this is the key that has a whole bunch of fancy words in it. And I just highlighted ones that people might not be able to define really clearly, or might have no clue about. And it's sort of a take home for me is when you look at a block of text like this, and I've highlighted all these words that are kind of bee specific, or they're Greek or Latin terms or whatever. If you're reading this, if you're trying to key something out, and you have to stop on half of the words in a paragraph, you're going to get tired really quickly and frustrated really quickly. And this happened to me, and I'm sure it's happened to everybody else who does this too. And before I even talk about some of those terms, I'll tell you that in Sandhouse, 1939, that same couplet, here's the drawings and the photographs that accompany it on the right. They're not there. They don't have those. And if you're lucky enough that there is a figure from a 1930 something monograph, it's going to be a poor black and white Xerox. So you just don't have the pictures that we nowadays have. So how am I supposed to figure out these terms without any reference? And so I took a few more couplets from the Sandhouse publication. And here's the words on the left that I thought, maybe some of these I would not have known what they mean. Most of them, I would guess if we had a test here amongst the audience, nobody would get all A's on this, I don't think. I certainly would not. And so this is what I was looking at as in my own frustrated efforts to key things out, accumulating all these words that I couldn't define. And it seemed fair that there should be one place you can go and get the answer. So that's what this whole thing is about. And here's a few more terms. It's interesting when you look at different keys. So the examples I have here are for osmium. And I had a bunch of words from there. And now if we look at a lasioglossum key, this is what's his name? McGinley's key. He'll use a bunch of lasioglossum kind of terms that didn't show up in the other one, but there's sort of within each taxon group, there's sort of a tradition of the sort of terminology that is used. And then if I go to another one, hold on, click here. If I go to an Andrina key, now I have a list of highlighted words that might be problematic, and they're all different, right? So you have to sort of, if you looked up everything and you're really rock solid on the Andrinas for a while, you go into a different kind of key. If it's old literature, you're going to run into new terms. And so as I have walked through all this stuff, I feel like I need help. And so I went to myself yet, which is what the whole glossary thing is about. So I'm getting to who this is for. Every non-taxonomist I know, this resonates with them. Like I haven't run into anyone who says, it's a waste of time. You just get out your Webster's dictionary and there's the answers. It doesn't seem to be like that. So I think that many of the people here need this. And I think anybody in the business, if you are rock solid, but you are creating resources for other people or teaching, you might want access to things like the glossary can provide for you. And part of what got me moving on this was working with Sam Droege, who many of you know, and Claire Maffei. They're the folks at the now semi-shuttered B Lab in Maryland. And Sam and Claire, they do a class. So I was attending their class regularly. And so I was watching other people run up against the same terms that I had run up against. And so Sam and Claire, they connected me with a lot of non-taxonomists, and they themselves know a lot, so they're the kind of people I want to work with. And it was with Claire Maffei that I started working on this glossary thing. And Sam gave me a title, which I really liked. So he called me a taxanarchist, which I defined because I get to define stuff now, as a citizen scientist that creates taxonomic resources independently of a funding source or organizational oversight, which kind of sounds like I'm rogue. So my next sentence says that, however, the best taxanarchists are all about collaboration and consensus. So I'll see if I can live up to that. So a little bit about how. It's really handy to be retired and be able to spend ridiculous amount of time on something that nobody else is providing money for. It doesn't fit into someone else's job description, but it's a niche that I think is worth addressing. And so I can just put 500 hours into this. So that's the first thing. And so with that time, I went to look up the problematic terms online and where are they referenced. And sometimes you can find a B reference. Sometimes you have to find the Webster's reference. It's like how does that fit when you're talking about bees? So I would find and then rewrite definitions to make them fit with bees, and either find pictures that people would let me use, or shoot pictures where I needed to shoot them to illuminate the question, the term that I'm trying to define. And my approach is to be as simple as possible, to use lots of pictures, to invite collaborators anytime I can, and I'm inviting anyone here to be in that group, and also to keep it light. And this one is for me important because, I mean, there's a psychology to this for me. I end up with a mild case of imposter syndrome when I am attempting to delve into taxonomy. It's like, I don't quite get it. Why can I not see that thing that I'm supposed to see under a microscope? Or is that scabriculus or is it scabrous? And I don't know. And so I feel like, oh, darn, like I just don't get this. So I want to try to communicate to other people. It's not you. This stuff is actually really hard to grind through. So let's not be too serious about it. So here's some examples, and I'll tell you sort of what the strategy is here. So, and by the way, I can also tell you, recently I moved the entire thing to a reasonable URL, which you can all remember. It's bglossary.org. So easy to find anytime. So Amanda will talk shortly about these awesome pictures. And here's the kinds of things I wanted to put in here and rationale for it. So some entries, they just need a sentence, right? So abscissa, I didn't know what it was, but apparently it's a piece of a vein between cross-sectional veins. So what I described it as, if Main Street was a vein, one abscissa would be a stretch between First Street and Second Street. Hopefully that's simple and not confusing, but it doesn't even need a picture, So another one is basitibial plate. It's pretty darn simple. It's a kneecap. So, I don't need to elaborate that very much. Decussate is also very simple. It just means they're crossed, but it seemed worthwhile to have pictures where you can, so this is definitely a decussate. It's a species that we see in the Pacific Northwest when we're lucky. And then this is like not quite. The mandibles down there are, they meet, but they don't really cross. And that's the kind of thing I want to be able to do, is to use pictures to clarify. Because, I mean, decussate is a really easy word, but how do you know it when you see it? And so it's really important to use photographs for that kind of thing. Gina, super easy thing to communicate. But if I can, I want to squeeze something in like here, where when you are asked to think about a Gina in a key, you're often asked to compare its depth to the depth of the eye. And so if you were just looking up Gina in a case where they're asking you to measure the Gina, here's a picture that suggests how you would do that measurement on an osmium. And then I have things that you kind of need a picture to explain. So an episternal groove in this picture here. There's a place where drawings, diagrams, or words work. And there's other places where you want to see kind of what you see yourself under your own microscope if you're trying to fool around with this. So for me, this is a very helpful image to get at this, the idea of where this groove occurs and what it looks like. Another one that needs pictures to explain for sure is when you talk about carinae, which are ridges of the propydium, and it comes up for a lot of different taxa, but I noticed it particularly for Lasioglossum. You want to see it big, so any of these images you can magnify them, and you want to see it like as clear as you can that here is the carina that we're talking about, and there's an oblique one and there's a lateral one, and then we can look at a different one here where this is very complete all the way around, and here's one where the lateral carina is almost it's very indistinct. But if you are struggling with looking at propodiums of lasioglossums, it's maybe helpful to see some pictures like this that, get you sort of in the right place. Another one that you kind of want to see pictures for are the humeral angle. Humeral angle is not a phrase that explains itself, so you really have to look at it. So here would be a humeral angle, so taking the head off, looking at the thorax from in front, and there's a smooth line here, so that would be, there is no angle, and here you can see a bit of an angle, and then here you can see this is about as angular as it could get. And then I have some entries that are simple, but kind of more fun if you can put photos in. And so these are the indulgent photos. I mean, I can define glossa in a word, but it's kind of fun to see, what it looks like on some real bees and how different it can look. And then when you're putting something together like this, you're lucky sometimes that you find something that's worth making a link to. Hold on. This doesn't make sense. Sometimes you get lost too. Let's see. Oh, here. This is pretty cool. So this is for that image where you saw one of these bees with a really elaborate glossa or tongue. And this is pretty nice work here. Which gets at one of the things I want to do with this glossary is not just have it be something where you need to know what something means and you look it up, but ideally, if I made this cool and interesting enough, you would just browse through it to see stuff that was like fun to see for taxa that you weren't familiar with. So there's examples of things that are, you don't need to know about gonadomorphs, you might not see them, but it's a really cool phenomenon where half of the bee is male, half of the bee is female. So you can see here's a male antenna, and here's a female antenna, and this one is really striking with a green sweat bee, where on one side here's you got that sort of female thing, and on the other side it's all male. So it's weird, and it's just fun and it's worth looking at. So I want to share that with people. And another thing I want to put in here, this is almost like an editorial, because I'm telling the reader that you probably need a reticle. And if you're not familiar with them, it's that in your microscope, you put this little plate in there and it has inscribed lines so that you can measure things. And maybe not even measure things in an absolute way, but get relative distances. So if I'm trying to figure out the length of the vertex on a bee, so this would be the back of the head to one of the ocelli. If you have a reticle, you can very precisely, if you're not shaking too much, which is usually the case, so you have to have your specimen really solid, but you can do the arithmetic on how this distance compares to the ocellar diameter. And so I want people to use reticles, because you drive yourself nuts otherwise trying to guess whether it's 1 or 2 ocellar diameters. And then I have some entries with links that are helpful. So it's not something I did, it's just something that is worth seeing. So this one was revolutionary for me. Bee mouth parts are super complicated. And so here's how they work. And that's just, for me, that's a discovery that I'm super happy having made, just for myself. And another place that I put a link in here, and this is also not something you need a definition for. Diagnosis is the word that we use when we're talking about trying to get the identity of something. But I think it's worth sometimes sort of, so we can toss that term off. What do we mean by it? And so this is another thing that I made where I'm trying to show the process of determining what some of the trait conditions are and running it through a key and getting some possible candidates, and then looking at close-up photographs to figure out whether whether the things that you found in the key are actually a match for what you're looking at. So it just walks through that idea of how you do diagnosis. Let's see. This is where I'm trying to keep things light. I really like this. It's indulgent for myself, but this right here is a tool that you get at the dentist for cleaning your teeth. It's a dental floss helper or something like that. And it has no place in this glossary except that the word denticle might be used. And anybody could probably see that refers to a tooth, but I just wanted to exploit it. So I said a denticle, a projection resembling a tooth, shaped like a tooth, or a dentist, a tooth care practitioner. And what I do with this is I say, ask your dentist for a few of these specimen cleaning tools. If you use this and you brush dust out of a specimen, it's a great tool to have. And so charm your dentist by explaining, I need these to comb my bead. And if they're not charmed, you should get another dentist. Another one is sort of trying to break down the intimidation factor of doing taxonomy is I have some text here about pronunciation of Latinized vocabulary. But basically, I'm saying, don't sweat it. Pronounce it with confidence. Then everyone else will think that you are all wrong. And a tiny personal example of this is, here's an agapostamen. And for the longest time, I called them agapostamen. And I kept running to people who said it, whatever it is, agapostamen. And it took me forever to convert to that. And if I'd taken my own advice, I think I would have at some point just pretended that everybody else just didn't know how to pronounce Latin and that I was right in the first place. Another category is things that require interpretation. And colors are really interesting. In older literature, a lot of times the colors are referred to with terms like this: fulvus, rufus, testaceous, pisceus, and so on. And I posted on a bee monitoring listserv where a lot of the old time folks who really know taxonomy, I posted sort of a request for people to give me some feedback on these color names. And this drawing in particular was annoying to some people. It seemed like a cruel thing to do to an Anthophora, but I'm trying to get the idea of like, what is ochraceus anyway? Like, it's pale yellow, I think. And if you ask a bunch of experts, which I did, they will give you very different but very precise definitions of what these color terms are. It sort of depends on who's asking. And an example of one would be ferruginous. So you can look it up and you can find, here I use the grammatical dictionary, botanical Latin, to name some things. But there's an editorial sort of thing I'm doing with my references to colors. I'm saying, okay, ferruginous is fine. Let's say it's rusty, iron, whatever. But why can't we just use something like brick red, burgundy, chestnut, auburn, etc. Maybe if I was writing a key from scratch in the modern era, I would skip ferruginous and go with rust. It's just an idea. And then one of these incidental things I ran across that's kind of interesting and helpful to me is I don't really know whether the precise shade of coloration is important, whether I need to use fulvus versus pink or whatever else as a color descriptor. But this was an interesting thing I saw where they showed how the pile on these two bees looked before and after exposure to the sun, which would suggest for this particular bee, that color is not a hugely useful diagnostic character. Another one that needs some context and description is relates to punctation. So, this is just standard, easy, straightforward. Here's these little, divots in the scutum of a bee. And this is the case of doubly punctate. So you have a large punctures and then all these littler ones. So you talk about the distances between, how dense are they? Like in the least helpful way, an author would say they're crowded or they're sparse. It's not helpful how sparse. So what what the best folks do, if you looked in Jason Gibbs keys, and a lot of keys nowadays would use this terminology where you talk about the diameter of the puncture in relation to the interpunctural distance. So you get an I over D value. So in this little example, I figured that it's about the interspace difference is about 1.5 to 2 diameters. It's getting a little wonky, but I feel like, at least for me, it's very helpful to have someone say, okay, how are they describing it and what does it look like? And I could add more examples of this to try to make it more clear. But anyway, there's that kind of thing. So back to, hold on. Okay, so those are some examples I went through. Okay, so far so good. I have this glossary, and it's pretty well populated. But I really want to have a morphology overview, because a lot of people, they're going to come into this that wanting to see the whole, like, how is everything connected to everything else? And the first thing I did was use images like on the left here. Oops. So just copies from Bees of the World by Michener, or from the Handy Bee manual, or drawings that were made by Rebecca Nelson. Just a mishmash of things. And sometimes these have been recopied for generations by now, because everybody uses the same kinds of pictures, and it works. But that's what I did initially. Then I'll explain to you how I got into phase two. Phase two was going to be, if I could start from scratch and I could do anything I wanted to, I would love to have original art. I'd like to see every perspective, dorsal, lateral, ventral, face, mouth parts, etc. It would be really fun to take something that's not just a generic bee, but it's an actual Pacific Northwest insect. And then I want all the labels to be hyperlinked, so you can quickly go to the definitions for them. And as long as I'm being idealistic about this, I want the images to be beautiful. So that was just like, yeah, sure. How are you going to do that? I am no artist. So hold on, click here. So then I was, just by fortuitous chance, I saw a message on the bee monitoring listserv, and it was for somebody who was trying to help somebody with an internship she was going to do scientific illustration, maybe interested in bees. And I said, "Oh, I need this person." And so I contacted Amanda, who was the artist, and I was ready to beg her to work with me, but she was so gracious, she just bought in right away. And so she did the drawings there at the top of the page. And I sent her a couple of specimens of Ceratina Mishneri, which is our model that we use. And then she did these things. Okay, Amanda's going to tell you about this.
There, let me get off mute first. Hi, everyone. I am Amanda. Thank you, David, for that lovely introduction to our meeting. And before I get into my spiel, I will say you're in very little danger of having to beg me to join the project. As soon as I opened the link that you sent and I saw the glossary in progress, I knew it was a great tool. And as someone who spends a lot of my free time looking at bees and looking for bees, and also someone who makes my living having to know these intricacies of anatomy, I knew I had been looking for this for a long time myself. So thank you for your 500 plus hours put into this. It was
You've done a few of your own for sure.
Yeah, I'm glad it's out there, and I'm so glad that I was able to contribute in this small way. So with that, I will get into it. I'm going to talk a little bit about the process of making these illustrations with y'all. And I'm just going to dive right in to beginning this art process. The first stage for me is not even usually art making proper. It's actually gathering references. And so what that consists of, in this case especially, is gathering existing high quality photography thanks to David, who had some of that on hand ready to go. It was a really helpful start to building this reference library. And then I'll go to scouring the internet, as so many of us do, for written and visual references for this species in particular, but also adjacent species for comparison. And so some of those included, of course, Bees of the World, Discover Life, et cetera, among many others. And while I'm doing that, I am making sure to cite and take notes all along the way so I can go back and kind of trace my resources to make sure I can use those in the future or double check myself for whatever I need to do. So to round out this slide, on the top here we have on the left one of David's beautiful photographs. And then on the right, there is a screenshot of my PureRef file for this project, which you can see looks a little bit chaotic, which surely I'm assuring you there is some rhyme in there for me. But one of the reasons that these files end up looking so chaotic is because I need ideally hundreds of photos per subject. And that's because I want to see the bee in its totality, but also be able to zoom in on each little part and see it from many different angles to really get a sense of how all those parts are going together. So after much study of this, I am able to pull out, a cohesive illustration in the end. Next slide.
Next one.
Yes.
Yep.
Thank you. Thank you. So from there, I will move on to study pages. I have all of my references organized, gathered in my file. So I really just start in my sketchbook making these series of smaller drawings that's just zooming in and out on this organism and trying to get a sense of who it is, how they work, all of that good stuff. So again, the purpose is to familiarize myself with the subject, and it is starting on a low pressure scale, which is very helpful. They're in my sketchbook. Typically, no one sees these. I don't think David even saw these before we put together this presentation. So again, working from that good photography, my own microscope photography, and direct microscope observation for these things. And in doing all of that, I realize what I think I know, what I don't know, and what I still need to figure out. So then I go ahead and figure that out by going back to the microscope again, back to the references, and finding additional things to fill in those gaps before I go back to the drawing board.
Next.
Yeah. Okay. So, 1 second. So once we have all those good hours in on the study pages, and I feel like I have a pretty good grasp on my subject, I will start all of that preliminary work. So at this point, those preliminary drawings were really trying to make a roadmap to final art. So two very important parts of that is composition, which in this case means taking into account also all of the space around the bee to make sure the labels fit in, and idealizing the subject just a tad, which involved kind of moving those legs around so nothing is obscured and you can see every part of that bee very clearly. And once I reach a stage where it feels pretty good, I'll send that back to David for revisions and, happily make those revisions if necessary and really try to get the preliminary drawing as accurate as possible. So once we reach that stage where everything looks pretty good, we'll go to final art. And if we do that preliminary work well, the final drawing is really actually much simpler than those previous stages. It should really just be a matter of refining the line, adding that texture and hair, and then balancing all of those things so nothing is taking up more attention than it needs to. And once again, I will email David and we'll make sure everything looks good. And then we can move to Illustrator, where we will begin to add in those labels, leader lines, again, troubleshooting the composition to see how those fit together. And David and I also put quite a bit of time into linking strategies to make sure that this flowed very smoothly into the glossary itself, which leads us to our last slide.
Okay. Oh,
Yeah.
is there another slide?
Oh, maybe I sent that too late. Well, it was
Yeah.
just some images of the final artwork, which you can see on the glossary. So,
Yes, we'll look at those in a minute again. Yeah.
yeah, so leads us to those final labeled images you'll see on the glossary, which are linked to David's beautiful entries, beautiful photographs. So it's a very time-intensive process. I hope that those illustrations kind of prove as a good starting point for the glossary. And if anyone has questions, I'll put my email in the chat and hand it back to David.
Yeah, and I'll be humble about myself, but not about Amanda. So I found the slides that she put into this presentation super interesting because I knew she was exacting and I knew she was working hard on this, but I didn't have any idea about how complicated it was. And I think Amanda and I both, we really wanted to be this species. And so I don't think she put in any extraneous punctures or wrinkles that weren't really there or hairs that don't belong on this Ceratina. And I think we made a good choice for species. In some ways, I would have loved to do an osmia, but they're too colorful and beautiful and hard to render in black and white, and they're too fuzzy. Most bees are too fuzzy. So that this Ceratina, it just conveniently, you can see the features that you want to see with it. But you also see, I don't say anything on the drawing. We don't have any links or discussion of the punctation, but we put it in there because it's beautiful and because it's real. So yeah, I was like super thrilled with all of this. So, okay. So what's next? My part is to edit and add things whenever I move to, but most especially if someone suggests what I might ought to edit or add. And something I found over a long period of time is that getting feedback is super hard. People say, "Oh, this is great, really like this." "Oh, tell me what you would like me to, adjust or whatever." And it's crickets. And it's frustrating because I have no doubt that some of the definitions I've done are not quite right. And another thing that I'm positive about is that the entire glossary is biased to my own experience. So there's a lot of Lasioglossum, Andrena, Osmia, Ceratina. Those are taxa that I have around and that I photographed. But there's not much. Like I was really pleased when Link Best gave me some of the Oregon, the key to genera of the Pacific Northwest. And I was thrilled because it had, oh good, I can use images that aren't ones that I already know, and they're different genera and so on. And of course, the glossary is also biased regionally. It's like all my examples are from Michigan or Oregon. And it's also, of course, it's biased to the United States. So ideally, it would be really sweet if I was able to add images or take suggestions from people in Indonesia or Patagonia, places where people think about bees just as much as we do and contribute to the literature and so on. So those are some things I would like to do. So my pitch to Bee World, here's a really easy way to condense that. Here's a little bit from one of those keys I showed you before. This is from which one is this from? This is from McGinley. So it's about Lasioglossums. And if McGinley had access to the internet at the time that he wrote his key, he could easily have put hyperlinks into his text so that anybody reading through this sentence that's going to choke on propodial, reticulate, scabrous, striate, scabriculus, pronodal, angle, what the hell is that? They could just click and go and find out, or ignore it if they already know what those terms mean. So my hope would be going forward that anywhere in the internet, when people are trying to describe bees or writing keys or whatever, if they don't want to spend half a page to describe what a pronodal angle is, and they can just link to the glossary, and maybe even tell me, yeah, I want to link to the glossary, but could you please adjust your text a little bit so it works better for what I'm trying to communicate, that would be great. So that's one thing I want to do. And then another thing that I want to do is promote to people who create web resources and who right now have some kind of a morphology overview set of images. There's no reason they shouldn't use Amanda's drawings. And the best way to do that I would give them a bit of embed code. So it would stick the drawings, and I'll look at her drawings again in a moment, but it would put those panels of drawings onto a web page wherever anybody else wants to use it. And the links come back to Amanda and me, so we don't lose control of it. So I'd be real happy if that were to happen. So when I say a couple of acknowledgements, a big one is I'm very grateful to folks who in the last 10 or 20 years have created new diagnostic resources that actually work for people who don't know what scabriculus means. That is, so I took a few samples here from the Pacific Northwest, key to genera, and you don't need to know a lot of terminology if you can look at the picture, and if what you're supposed to see here is a strong carina compared to another bee that doesn't have a strong carina, you don't even need to know what carina means. The picture is telling you what you need to look at, right? Or same thing for a pseudo-pygidial area. You don't need to know what that is. You're looking at your bee and saying, does it have a little line or doesn't it have a little line? So that's the trend for new resources, and I applaud that. I also like the key to the megachile, where same sort of thing. This one's dichotomous, and it's just the little arrows tell you where to look on the mandible to compare this set of teeth to this set of teeth. And then I'm always also very grateful to Discover Life, which, if you're not familiar with it, is it's not dichotomous. They have matrix keys, and the intention in Discover Life is to make everything in plain English as much as possible. I'm still weeding out lots of terminology that's complicated there, but the spirit of Discover Life is to make it so a citizen scientist can work through it without having to take a degree in entomology somewhere. So, and then of course, I could acknowledge any contributed. This is not exhaustive, but I mentioned Claire Monte at the U.S. Fish and Wildlife Bee Lab. Lawrence Packer was very generous in letting me access his photographs. I just mentioned Discover Life. Lisa read through, Lisa's a way better editor than I am, and so she flagged all kinds of embarrassing typos, and maybe she'll have to do that again sometime soon. Recently, Link gave me images that were shot by Joshua Dunlap, and that was helpful. And then all kinds of resources, Missioners Bees of the World, I could go on. And last but not least, of course, Amanda. So that's my whole thing. And I'm going to go back to, hold on a second here. I want to go back to the web page and look at the pictures. So when you look at this yourself, the way these pictures work, the picture is small on a web page, and it's smaller if you have a smaller screen, even in this. So they blow up. And so you can, one at a time, you can look at the lateral view or the dorsal view, and then you can click and jump to the part in the glossary where that term is discussed. So that's how that works. But for me, again, I can praise Amanda as much as I want and be humble on my own behalf. This is a game changer as far as I am concerned. If someone can point out to me somewhere where someone has done a better job of this, great. All hats off to them. But there's nothing like this anywhere else that is as thorough and deliberate and beautiful, in my view, as these drawings are. So that's the whole deal. And I'd be really happy if there were any questions or if things came up in the chat that I should address.
People pretty much answered as we went, so
okay,
it worked out pretty well, actually. It's really beautiful. And I think we should all clap and stand up and give you a standing ovation at this point, which you couldn't see. Lots of people say, nicely done, psyched to use this. Oh, Erica said that taxonomic keys are mostly dichotomous and most characters are based on presence or absence. But when new species are described, we'll need new morphological elements. So if you're adding new species, please add those and tell us what they are and get them in there.
Yeah, yeah. I can't emphasize enough that this whole enterprise, because I'm just working by myself in my house, is based on my intuition about what needs to be explained, and it's narrow. So I'm excited for anybody else who can tell me, would you please explain these weird things about nomia, which I've never seen. I only know they exist, or whatever. So
yeah, there's some
please hit me up.
hind legs out there in Nomia land. Kit Pendergrass was mentioned a number of times. Have you met her from Australia?
I have not. No.
I'll get you together. I have her contact information. And Amanda also has some amazing pictures I somehow managed to find. And there's a really beautiful squash bee out there in Instagram.
Thank you.
I'm going to make one other comment about the drawings, which is like I do a lot of photographs, right? And I do focus stack pictures of little tiny parts of bees, and sometimes they're really helpful. They're helpful to me, but drawings, it's an alternate way of seeing something that's super useful. So I have photographs in the glossary of an Aurolium. It's not nearly as beautiful as this or as distinct. So when you draw things, you have the options. You don't have to worry about the light. You're in control of that. You get to decide how heavy the line is going to be or not. And so you can make things very clear that are hard to get in a photograph. And I think the goal, if you're doing anything like what we're trying to do here, is use photographs, because that's what the user sees through a microscope. So if anybody has done like cell biology classes and so on, there's the mush you look at through a microscope, and then there's beautiful drawings in your textbook of exactly what you should be seeing. But no, it doesn't look like the drawing, So you need both. The drawing sort of directs you to, in a very efficient way, to the key things you want to see. The photograph, it says, oh yeah, okay, now I can see that stuff the way it looks in real life. So
yeah,
that was helpful to me to think about that.
It really makes a difference to see a photograph next to a diagram. There is a question. Do you have any idea if there's a term for the tufts of hair some Andrina have that are above their eyes that look like little eyebrows?
What kind of species, for example, are we talking about?
Andrina.
Yeah, a species of Andrina.
Oh, Haley, would you like to pop in?
So like I have this a small promotional piece here. I have this other thing that I call Andrina Deconstructed, a guide to the characters. So I've looked at Andrinas a whole bunch, and whoever asked the question, if we scroll through here and you see what we're talking about here, let me know.
The fovea, these little white patches, or do you mean something else, Haley? Oh, she's saying it might be the facial fovea. I Googled that and I think that's what I meant. Okay.
Yeah. If you look at my Andrina guide, you'll see lots of different pictures of fovea. But that's the only thing that rings a bell. As for you, Lisa, if you're talking about hairy stuff on the head of an Andrina, the only thing that's unusual and distinct for Andrina in particular is the fovea.
Yeah.
So did you build the website and all the hyperlinks and everything too, David?
Yeah, that's a part. I won't make you think about this, because it's really not very interesting. But this is like the code, right? And it's just that it's an exercise in masochism to try to get this stuff to work out, because so you're probably familiar a little bit with HTML and CSS and JavaScript and all that. And nowadays, you want to make a website and make it jump through certain hoops, you have to do all this code stuff, So I learned about it. And I'll make a confession. I say in the about page for the glossary that the entire thing was done without any kind of AI, because I'm on guard about AI. In B world, we've seen some cases where it's used, to actually create content, and it horrifies me, and it's usually really low quality. But
Yeah.
I use chatbots to do code for sure. It's the only way I can do it.
That's interesting. Where is your Andrina guide located so people can find it?
Well, it's linked in the glossary in a number of places. So if you just, that's well, it's also, I mean, its URL is bglossary.org, right? If you type in bglossary.org, it takes you to the glossary. If you type in bglossary.org/Andrina_guide HTML, that's the Andrina page.
Okay. I find it by looking up your name.
Yeah, that should work. Should work too.
That goes there.
Yeah.
And he also has a little Serotina key there.
Yeah, and let me just say one other thing, which is this is sort of a philosophical thing. So I made this glossary thing, and I made the Andrina guide because I was frustrated, and the resources that were out there weren't sufficiently precise enough for me to learn as fast as I wanted to learn. And so I became a taxonomist, right? I just started making stuff. And I really promote that idea. Like, anybody can do this, right? I mean, not everybody has 500 hours they can dump into something, but you can chip away around the edges in some fashion or other. And I'm sure Washington, your Bee Society, a lot of you are doing that very sort of thing. But the idea that ordinary folks can do taxonomy is attractive to me, with the giant caveat that it's taxonomy, identifying things. It has to be, to have an 80% sure that it's species X is really not very useful. I mean, it's useful in the process of learning, but it's not useful in terms of adding to the knowledge we have of bees. We need to know what they are. When we say there's something, it needs to be that thing. So there's a real hazard in me saying, anybody can do taxonomy. Shucks, I didn't study it. I didn't train under Jason Gibbs or whatever. I can just do it, but I can only just do it because I've spent hundreds of hours doing it. It's hard, but it's doable.
Yes. Okay, we got a couple more comments or sort of questions. Can you think of any of other instances in which an experienced science illustrator could make a big difference in clarifying information and making it more accessible to a wider group of people? Have you suggested hiring an illustrator for something specific to any of your colleagues, for example?
Okay, well, this is definitely in Amanda's world. Tell us, Amanda.
Yeah, there's a lot of instances in which I think science illustration can be a really useful tool. And like David said, I do think that this is a really good example of it being at its most effective, where you have tons of photographic examples next to illustrated examples, and together they kind of coalesce to give you the best possible picture, or best possible starting place to understand what you're looking for or looking at. But beyond that, science illustration can help clarify really complex processes, or it can tell more of a story. So a lot of my work also focuses on, life history illustrations, which is telling you maybe the story of an insect from its birth to its death and all the in between, and then also like who, what other organisms are relating to it within that. So that can be hard to tell, in one or a series of illustrations, but or photos rather, but an illustration, you have the power to kind of play with time and play with space and idealize all of the settings, and stuff like that, and also very microscopic things. Sometimes that's science illustration can be helpful as well.
Yeah, just one comment I would make about scientific illustration is, like, right now, I'd say in bee world, there's hundreds of people who do really good photographs, and not very many people at all that do really beautiful drawings. And of course, 100 years ago, it was the other way around, and if you want to see really great scientific illustration, that does what photographs can never do. You should look at things that are 100 years old. They
Mhm.
did amazing things in those days. I mean, even if you just think about Audubon or whatever, you know, but there's a beautiful, rich history there, and I think it's a resource that we don't have enough of in sort of scientific exposition, in taxonomy. You know, just trying to communicate about the beauty of things. Drawings, we don't have enough drawings.
And this is a question for Amanda. Do you teach anywhere? Or do you have plans to teach anywhere?
So right now I do teach workshops sometimes. I don't have a full-time teaching position, although it would be something that I would be interested in the future. Right now I've got enough freelance stuff on my plate where I don't quite have to take on a full-time teaching position
kind
yet.
of
But
froze.
I do like teaching. I enjoy working with students, and
I would really love to see this, you know, go down to like a high school level, getting like high school students interested in something like this. Was, you know, yeah, I mean, it would be like one or two would just go, this is what I want to do. And like, you know, I
right.
mean, you get them at like 13 or 14 or 15 and like, you know, that's, yeah,
Absolutely.
I have your contact information. We're going to talk.
Yeah, we'll talk. I agree.
Excellent.
Especially, you know, I didn't know that being a specific science illustrator was even a career until a few years ago. I studied general editorial illustration, and that was the world that I was working in before I had my insect awakening. So you know, getting to those kids young can make a big difference.
Mhm.
Have you worked with flowers at all, or
What's that?
have you worked with flowers, or
Oh,
because that
yes,
would
a lot of plants too. Yeah, for sure. Lots of plants.
Yeah, actually, I'll share that what Amanda was telling me recently is that she's taken on this super cool niche, which is gall insects, right? The things that, I mean, in the Pacific Northwest, you see oak galls all the time, but there's a zillion kinds of galls. And somehow she's interested in the little tiny squirmy things that are in the middle of those. So stay
Yeah.
tuned for whatever she
Awesome.
generates.
The
Those,
outside. There's a
yeah.
question for Amanda. Do you
Oh.
ever do illustration for areas outside of biology? E.g., I'm a physicist and we could use better ways of communicating.
Yeah, I will say that my wheelhouse lies within insects and plants and the animals and other organisms surrounding them. But it benefits me to be good at many things, so I would be open to working through physics material with you or with, you know,
And
whoever might need
and
them.
there's an ask for a glossary for grass species and suggestion back there. Let me go back.
That's a whole nother thing.
Yeah,
That's a big one.
I'm out on that one.
Grasses are hard.
Yeah. The links from the illustrations to definitions are great. Could there be link backs from the definitions to the best instance of each term in the illustrations? So
Ooh. Yeah, it's boy, I don't know. I don't know. I think it's pretty hard to go back.
yeah.
Like if there's a particular term that's in the drawing, meso, plura, or something like that, in the definition, I'm going to want to probably, if I commit some time to it, say something about meso, plura of things that aren't Ceratina, like things that are going to, right, like Ceratina, it's great for the general map of a bee, but it's just one kind of bee. And so in the body of the glossary, I think I want to try to go as broadly as I can to look at, you know, every other kind of taxon.
To you, Amanda, Chris Looney, who's our Washington Department of Ag guy, is a big time gall person.
Oh, really?
Yeah.
That's exciting.
Yeah.
Yeah, I might ask to get in touch.
He's a Washington Department of Ag, so Dr. Chris Looney, Looney.
Okay, great.
And David, if you have a
Yeah.
link, you can send it to her.
Yeah, I can connect you with Looney too also.
Fantastic. Yeah, I'm in the process of gathering, you know, a bunch of research for a gall project a personal project that I'm working on, so it would be a good time to reach out.
Yeah, he's been into galls for a long time and gall wasp, and he's put on a helped put on a couple international seminars around it.
Oh,
Ooh,
fantastic.
Plug into him.
I
does
will.
anybody else
David,
have another question? These guys are, I don't know where you live, Amanda, but David's back in the middle of the US and it's much later there.
Oh yeah, no, I work until forever, so it doesn't matter. I could go on for a long time. Yeah, don't tempt me.
Okay.
I'm in Auburn, so I'm 1 hour behind David, but happy
Okay.
to hear. Oh, and just to add to the Saratina conversation, the folks here at Auburn say Saratina,
Oh,
which I've never
Oh.
heard before, but now we have
Oh.
another option.
I like,
Something. Go ahead.
I was just gonna say, I like what my Don Rolfs was my mentor, and what he said is whoever says it first, everybody says it that way, which is a kind way to go about it instead of
As
arguing.
long as we all know who we're talking about, I'm okay with it, you know.
And Karen Wright says it's a dead language, so say it however you want to.
Yeah, good. I'll
So,
quote
David,
her in the glossary.
Are you going to expand beyond Andrina? Are you going to be doing other ones?
Well, that's a good question. So that Andrina page that I have, like, I think it's brilliant, but that's just me. What I mean is, what I do in the Andrina page, but for one, I just walk through. Here's what we mean when we say it's got a slight humoral angle, blah, 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 sort of interpreting things. But then there's those diagnoses. And I would love to see a page of diagnoses for other taxa. So like what I do for Andrina is I take a specimen that I have and I photograph a bunch. And then I say, walking through a key, sometimes I nail it. A lot of times I say it could be this or it could be that, but I'm not really sure. Go back to the original literature. I'm still not sure. But I think it would be really useful to lots of people. It'd be useful to me for somebody who is good at osmia or good at at megapyly or whatever, to do a systematic walking through. Here's how I navigated each couplet in a dichotomous key, or each trait condition in a matrix key. I think it's empowering for people to look at how you go through it, and also sometimes seeing that you don't get the answer and that's just the way it is, because you're not. I mean, sometimes we don't get the answer. The experts don't always get the answer. But I'd like to see more resources that are about particular taxa. How do you tackle this one? Like for osmia, I mean, I would love to have 2 pages worth of awesome pictures of mandibles, because they're so important, and interpreting them is difficult, So yeah, I'm open. I need collaborators, and I'll jump on anything.
I want all the masks of the male Hylaeus like they do in Hawaii. There's a diagram with all of them in black, and they're illustrated black and white drawings, and just because if you could just see the shapes of them all together, you could go, well, it's not that one, it's not that one, it might be this one, it might be that one, and you would have a head start. Yeah,
that's good. Well, we need all kinds of different ways of tackling this thing, and my general mission is how someone who's not a card-carrying taxonomist can contribute to the literature and to the general knowledge. And I think knowing things to genus is not very helpful. It's very helpful in terms of getting excited about what you're seeing. It's very helpful for certain things, but you don't know anything biologically until you have a species name. So it's an important thing to be able to do.
The photographic and illustrations are excellent. Can you both connect with Washington and Oregon Bee Atlases to help illustrate interesting and hard to see characters? We're growing all the time and getting better. So that's an idea for the future, I think. And when you have 600 species in your state or more in Oregon, that's a lot of things to illustrate.
It is, yeah. I mean, I'll speak on Amanda's behalf. After her most recent set of drawings, I tripled her compensation. I paid her 3 times more than I paid her before, which was, I don't pay her anything. So it didn't really make any difference. So I wouldn't presume that you're going to be able to get Amanda, just because she loves bees. I'm sure she's got a million other things she's got to do, and she's got to pay the bills too. In some ways, if you're looking for like free stuff, I'm open to it. I'm not making any promises, but if somebody in Washington or Oregon said, like what I just suggested, if you said, let's do a little sort of tutorial on osmia, you sent me a bunch of osmias and we went back and forth about it. We could create a web page about osmias that would be helpful.
Anybody else have a question?
Oh, I guess I just have a remark. I guess that that glossary is not exclusively handy to use in bees. I bet it might actually have some value in IDing apoid wasps. So maybe if I ever have a term that I'm confused in, I'm going to see if that also helps with other apoids too. Because
yeah, there's a Hymenoptera plural, like a giant list of Hymenoptera terminology that doesn't have all the pictures and so on, but it has specific descriptions. I was a little surprised at how many of the terms in the glossary are just bee terms. They're specific to bees. It's a little surprising. None of those terms would be applied to a syrphid fly. They have their own nomenclature, unfortunately.
It would be nice if it was sort of kind of across all the different bugs, so you could just go, this is this across, if it's an ant or a fly or a beetle or whatever.
Yeah, it mostly isn't. But another thing that's worth talking about for a second is surface sculpture, right? So when you talk, and it's really important because whether bees have like complicated rugae, or whether there's little scratches in the integument, or whether there's little pimples, or you could use common language to describe the different ways that sculpturing occurs on the surface of a bee. And lots of people who want to talk about that in any detail reference the paper that was done on ants in 1969, and it has electron micrographs of different surfaces of ants and a very specific dictionary of terms and exemplars of what those are. And Bee World needs that too. Like, it would be helpful if we all understood what scabriculus meant, or maybe there's a better term for it, but if the terms were somewhat standardized so that, as it is for ants, we know what we're talking about when we say rugulos instead of rugose, right? I mean, where's the dividing line? And I didn't have the time to do it, but it would be fun to make some type of a document that said, superficial impressions, here's all the names for them. Excavations, here's all the names for those things, and here's some examples. Like, that would be a fun thing to do, and it would potentially be useful, I think. And
people that are still learning bees to genus, just keep looking at wings. You got to start somewhere, and you just go with the ones, like he really dove into Andrina, and you go with the ones you love and you want to work on, and see if you like the stuff that Thomas Onufurku has done on photos for Epiolus and Triobolus have been really useful.
Yeah, he does a good job in presenting stuff. So last thing I can think of to say is that I live in Michigan. It sort of breaks my heart that I'm not in the Pacific Northwest, because I became quite fond of the taxa there. Some of the people too, I must say, but it's tough not to see the same species that I was used to seeing.
We have a few more. When there's like 700 across the whole of the eastern US, it's kind of strange to think about.
Yeah, you have more. Yeah, it's all Bombus impatiens here.
So basically, we have almost as many species in Washington State as there is for most of North America, or the rest of
Is it east of the Rockies? Does anybody know? It's 700 and something. It's like they only have one hummingbird, and I have 4 at my house. Lucky, we have a lot more diversity of eco regions. We have what, 57?
Yes, that's what it's about.
David Jennings, 57 in Washington, level 4 eco regions, which makes a lot of diversity too. I think it's time to wrap it up and let you guys go. Thank you very much. This was very entertaining. Lots of good, deserts are great for bee diversity. That's part of it. We have about half of all the bumblebee species in North America and Washington. So people are adding comments right now. Look forward to seeing more of your work. Happy to read through it again. And anybody else, if you're missing a term, please tell David, we'll add it. Or if you need an explanation of a term, you're still not getting it after you read it, and laugh at his jokes. Tell him they're good. Makes him happy. That's good feedback, so it keeps going. Thank you very much.
All right.
I appreciate it.
Thank you so much to everybody.
Let's say good night.
Good night, everyone.
Nice